Good afternoon, everybody, or good morning, depending where you are. And thank you for coming along. So yes, I'm going to talk about Fusarium diseases today, a major challenge for diagnostics and plant breeding. So this is just to introduce some of the work that we're doing in my group here at Warwick. Okay. Whoop. Okay, so yeah, I'd have to get the obligatory bicycle picture in my introduction, but anyway, this is just to let you know what's coming up. So I will give you a bit of an introduction to Fusarium diseases, specifically Fusarium oxysporum. And for this afternoon, I'm going to focus primarily on onion and lettuce as exemplars of some of the work we're doing. Necessarily, because I'm going to be talking about diagnostics, I'm going to give you some background on these pathogens and also a little bit of genomics. So be prepared for that. So yes, on the diagnostic front, I'm going to be focusing on Fusarium basal rot in onion, particularly highlighting a current project we've got called the Fused project at the moment. And in terms of resistance, I'm going to be focusing on the lettuce side of things, on work we've done on identifying new sources of resistance, but also importantly, characterization of new races of Fusarium in lettuce. So more about that coming up. Okay, so here's my standard introduction to Fusarium oxysporum. So it's a globally distributed soil-borne fungal pathogen, and it causes a variety of different vascular wilts and root rots. And if you look on the right-hand side there, those are some of the symptoms in some of the crops that we've worked with in terms of Fusarium diseases. And on the text on the right, on the left-hand side, you'll see Fusarium oxysporum can infect a wide range of crops. The ones underlined there are those that we've worked on currently or in the past. But the key thing to remember about Fusarium oxysporum is that it's a species complex. So it has these different forms, which are known as forma specialis, and these are uniquely adapted to these individual hosts. So that's at least 120 different forma specialis. As I say, they're all adapted to different hosts. Just to add to the complexity a little bit, there are races of these individual forma specialis that can evolve, and that's in response to resistant plants, resistant cultivars being developed, and then perhaps predictably in some cases the pathogen can evolve to break that resistance. So I'm going to talk about that in the context of lettuce later on. Also, Fusarium oxysporum notoriously difficult to control, being soil-borne obviously, but it also produces chlamydospores which can survive in the soil for many years. And interestingly, within this sort of complex of plant pathogens, you also have non-pathogenic forms of Fusarium oxysporum in the complex. And these are abundant and pretty ubiquitous in soils. So you can see having this species complex as a whole, including non-pathogens and pathogens, this does make diagnostics rather challenging, as we'll see. Okay, so just an introduction to the main pathogens I'm going to talk about today. So firstly, Fusarium basal rot of onion and garlic. It's caused by Fusarium oxysporum forma specialis CP, which I'll call FOC for short, going through. And it is pretty much present wherever onions are grown, and it causes this basal rot, as you can see in the photo of the onion, this internal rot. So the fungus comes up through the roots, infects through the basal plate. And in the UK, at least, it wasn't really common or really not widespread before it was first identified. We think around the 1990s, so even before my time. But since then, it's definitely emerged to produce increasing problems in onions. So now it has a pretty high impact, with losses estimated around £20 million per annum. So the plants can become infected in the field. So you can lose plants in the field, but one of the main problems is plants, or bulbs rather, that look apparently healthy that then develop the disease in stores. So a farmer can harvest the bulbs, put them into store, and then they will develop basal rot. We probably don't have really, complete sources of resistance to basal rot in onion, but there is partial resistance out there. And so one of the priorities, at least, that we've been dealing with is to develop diagnostic tools to assess disease risk. And as we'll see in a moment, that really is focused on, or at least one component is focused on, a soil test for this particular forma specialis. So the other pathogen I'm going to focus on is Fusarium wilt of lettuce. It's caused by Fusarium oxysporum forma specialis lactucae, or FOLA for short. Again, this causes wilting and plant death in the field. That's the main issue in this particular disease. And this is one of the forma specialis that does have races. So we've deployed resistance in the field, and then the pathogen has evolved to overcome that resistance. So there are 4 known races. Race 1 is widespread, particularly in warmer areas of the world where it's present in field lettuce. Race 2 and race 3 don't seem to have advanced much further than Asia, at least they're not common at all outside that we know about. And of interest to us in particular in the UK is the emergence of a new race, race 4, or FOLA 4 I'm going to call it, which is pretty newly emerged around 2016. And it's also fairly new in Northern Europe as well. So before that, lettuce growers didn't have a problem with Fusarium, but this new race occurs. It looks like it infects at lower temperatures compared to race 1. And another issue is that it looks like these races are continuing to evolve. So we've been doing some work on identifying these new races or variants that are beginning to emerge beyond the known races 1 to 4. Okay, so for Fusarium basal rot of onion, as I said some time ago, it really wasn't that common, but this is some data from Andy Richardson at the Allium and Brassica Centre, and you can see this is the percentage bulbs affected that they were finding in stores over the years from the early 2000s through to the present day. And you can see certainly from about 2012 onwards, there was quite an increase in Fusarium basal rot. So it is probably the major disease that is faced by onion growers now in the UK. Going back to lactucae, again, I mentioned this new race, so race 4 spreading in Europe and the UK, where previously Fusarium in lettuce wasn't a problem. I'm not expecting you to read all this text, but this is a kind of timeline, if you like. And I've just highlighted a couple of instances there in red. So 2015 was when race 4 was first described. So it's first identified in the Netherlands, and subsequently in Belgium. And for Ireland and England, we first identified that race 4 in 2017. So that obviously caused quite a lot of consternation at the time, because growers really didn't know what they were dealing with. Since then race 4 has continued to be identified in other parts of Europe, including Italy and Spain. And we also see reports of race 1, interestingly, creeping into Northern Europe as well, albeit in indoor production in Norway and Northern Ireland. So it's a continuing sort of evolving picture, if you like, of the spread of race 1 and race 4 into Northern Europe and the UK. So the current status, as far as I know, at the moment at least, is race 4 in the UK. There are multiple growers affected, but it is, and it's worth stressing this for the moment at least, it's confined to indoor protected production. and we're not aware of any outbreaks in the field, but happy to receive any suspect samples if you think it's in the field. It can cause very high crop losses, up to 100%, and the varieties affected, certainly at the time of the outbreak a few years ago, with the little gem and butterhead type. So since then, actually, the seed industry has moved quite rapidly to bring in, or start to bring in, some race 4 resistant varieties. Okay, so that's a brief introduction to the two pathogens I'm going to focus on. So I'm going to now talk a bit about how we are developing diagnostics with the example of Fusarium basal rot of onion. So I have to give you a little bit of background on this to give you some idea of how we approach this. So we're talking molecular diagnostics here, and I guess these days it's routine to sequence what we would call a barcoding gene, such as the ITS, or in this case the TEF, the translation elongation factor. It doesn't really matter exactly which gene we're talking about. And then sometimes you can develop a specific PCR from the differences in that sequence. Now, the problem with Fusarium oxysporum is it is a species complex, and they're all very closely related. So we find that if we sequence the TEF, we can distinguish different Fusarium species quite well. So you'll see in that family tree of Fusarium some other species at the bottom there. But in the top half, we have the Fusarium oxysporum complex, and we find that sometimes we get identical TEF sequences for these different forma speciales that affect different crops. So really, that's not a great approach then for diagnostics. So that was the kind of challenge we had some years ago. These days, and it's becoming far more routine now, is that we can actually go and sequence genomes, and we've been doing this for some time in collaboration with Helen and Jordan at NIAB. And it's worth just pointing out some features then of the Fusarium oxysporum genome, and it's, if you like, compartmentalized into two parts. So firstly, we have the core genome, and this is sort of a very sort of rough cartoon to demonstrate this. So the core genome is highly similar between different forma speciales within Fusarium oxysporum, and it contains genes that do the general housekeeping functions, including those sort of housekeeping genes or barcoding genes I mentioned previously. And it is really highly conserved. So there's really not much room there for identifying diagnostics based on that part of the genome. However, these forma speciales also generally have an accessory genome, as it's called. This can be one or more mini chromosomes, and these are actually highly variable between the forma speciales. And it's really this area of the genome which is where all the action is, because it contains the majority of the pathogenicity genes, so the genes that allow Fusarium oxysporum to infect these individual hosts, and therefore it largely determines the host range. And therefore it's this part of the genome that we really need to sequence and get data on if we're to develop diagnostics. And this is what we've been doing with Helen and Jordan. So, and it's quite difficult actually to assemble these parts of the genome. So we've done that. We were first to publish both the genomes for Fusarium oxysporum CP and also more recently for FOLA, so Fusarium oxysporum lactucae. So if you're excited about that, you can go off and read those publications. So as we begin to sort of delve into this genome then, and into the accessory genome in particular, we can run these bioinformatics pipelines, as they're called, to identify these effector genes or pathogenicity genes in different forma speciales. And this just shows you, if you like, a presence-absence of what we think are pathogenicity, the main pathogenicity genes in these different forma speciales. So you can see a few isolates of lactucae race 1 and race 4 at the top, and halfway down in yellow is the CP1. So you can see you know, some of these forma speciales have some of these pathogenicity genes in common, and you can also pick out unique genes as well that differentiate between the forma speciales. So you can see from this sort of diagrams, then you can pick out targets for diagnostics, not only for the different forma speciales, but also potentially for the different races within the different forma speciales as well. And as a sort of side note to this, there's one specific class or type of pathogenicity genes which have kind of become quite popularized within Fusarium oxysporum. These are the secreted in xylem genes, and I guess they're popular because they're a bit more well characterized than many of the others. So you can look at these in isolation if you want. It's best to look at the whole genome, but again, this serves to illustrate that for these 14 known secreted in xylem genes, which are first identified in the tomato version of Fusarium oxysporum, you can see there's different complements of these 6 genes. So I've highlighted CP, which is towards the top here, and it has, you can see these red boxes indicate it has 7 of these 6 genes, if that makes sense. And lactucae, we've got a couple of the races, race 1 and race 4 I've highlighted. So race 1 contains 6, 9 and 614, and race 4 also has 9 and 14, but has also 6, 8. So hopefully that gives you some sort of background to where we're coming from about developing diagnostics and how we can begin to distinguish forma speciales and the races within them. Okay, so moving on to sort of more practical applications, then what we wanted to do was develop a diagnostic for FOC in onion. And so we did this based on one of the 6 genes, and we developed a specific quantitative PCR test. We tested it against lots of different Fusarium species and other forma speciales and found it was quite specific. So that was good news. And we also, because we have a quarantine field here, which we had inoculated at quite a high level with FOC, we could extract DNA from that soil when onions were grown in the field. And you can see from this graph, this just shows you the readout of the pathogen DNA over time in our quarantine field that's been artificially inoculated. So you can see we start off with quite a low level, and then when onions are grown, perhaps predictably, we see an increase in signal as the pathogen proliferates on the onion crop, and then we see a decline after. So all good, and we thought we'd done a good job there. But when we actually went to use this qPCR and test it on grower fields, we couldn't detect FOC pretty much anywhere. So we concluded that really this PCR test really wasn't sensitive enough. So we needed some way of increasing the sensitivity. At the same time, just as a footnote, we did develop PCR diagnostics for other forma species I'm not going to talk about today, but Fusarium on lettuce at different races, and also Narcissus amongst others. So the key problem then was how do we use this qPCR test to practically detect FOC in onions? One thing we did show, though, before we get on to increasing the sensitivity for the soil test, was that we could detect Fusarium in asymptomatic onion bulbs before storage. So you can probably identify the sort of rotten bulbs, so you wouldn't be putting those into store. But the problem comes, as I said earlier, from bulbs that look apparently healthy that then develop, so they're latent infections essentially, which then develop in store. So we did show, for instance, and this was just an AHDB funded experiment we did. So we took 200 healthy bulbs that did come from an inoculated area, but they look completely healthy. And you can see the basal plates in the photographs there. We extracted DNA from half of those, so from 100 of those, and we detected Fusarium in 50% of them. And then if we put them in a nice warm environment, or the other half of that batch in a nice warm environment, so hot boxing it's called, then we saw that Fusarium did in fact develop in those bulbs. But industry-wise, it's probably not practical to do that, that scale of testing on onion bulbs, given the time scale between harvesting them and putting them into store. So our real focus then became to try and develop this DNA-based molecular diagnostic as a soil test, so to increase the sensitivity. And this is where this FUSED project comes in. So this is a project that's been running about a year and a half now, funded by DEFRA and Innovate UK. And as you can see there from all the logos, it involved major players in the onion industry. So there were 4 different objectives to this particular project, which is ongoing. I'm not going to talk about all of them. I'm going to take a Warwick focused view, I'm afraid, here. So talk about the ones we're involved with. So mainly the soil diagnostics, which is the first one. But just to flag up that there's some agronomy work going on in this project as well to understand the agronomic factors that are exacerbating Fusarium basal rot expression and look at management options in the field. The project's also headed up by Beehive. They're interested in developing a computer vision system for early detection of infected onions through the sort of crop growth cycle and into the harvesting pathway. And finally, the last part of this project is to look at a volatile gas detection system, which we're also working with at Warwick, and this is work led by James Covington in engineering. And this is the idea is to implement some sort of sensor unit to detect these volatiles associated with infection of onion, to detect the pathogen in store, particularly during the sort of curing and drying phases. So I'm going to talk a little bit about 1 and 4 here. So as I said, the major challenge really for us was to increase the sensitivity of our molecular diagnostics. And so the way we went about doing this was really leading on for a publication that was associated with the lettuce pathogen. And this was to, before extracting DNA, so rather than extracting DNA directly from the soil, we actually add the soil in to a Fusarium selective media and incubate for a bit. So if there's any Fusarium spores in there, they germinate and grow a bit, and therefore essentially you're going to boost the amount of pathogen in your sample. So we then centrifuge that, and then we can extract the DNA from the soil pellet and what was growing in the media before we run our PCR. So we call this enrichment. And so this enrichment did indeed allow us to increase the sensitivity of our test. So we did some very simple experiments here where we can spike f.sp conidia into the soil. This is in 0.5 gram samples. And you can see here from the graph on the right-hand side, these, the smaller bars, the red bars, represent detection using direct DNA extraction just from soil, so without enrichment. And we can detect down to about 1,000 spores per 0.5 gram of soil. Whereas with the enrichment, we can go to at least 10 spores within that sample, or even less than that. So we were confident then, at least from a lab point of view, that we had increased the sensitivity of our test using this enrichment. We then tested this in different soils that onions might be grown in. And you can see different soils along the bottom of this graph. And again, these columns represent different numbers of spores spiked into these different soils. So you can see a nice dose response. And you also might notice that the black soils or the peat soils, we get slightly reduced signal. And those soils are well known potentially to inhibit PCR reactions, but in this case it's not too bad. We can still detect the pathogen quite well. So we then implemented this enrichment protocol to real-world samples from a number of different onion growing soils from around the UK. So what you're looking at here on the left-hand side is samples that were taken pre-planting, so before onions were grown. And along the bottom there are 21 fields, and you can see that we can detect FOC in the majority of those samples and at different levels. We then went to the same sites at harvest time, and you can see we get pretty much the same result, although we do get a few extra field sites lighting up. So it does look like this test, this enrichment protocol, does have some potential then to determine the amount of FOC pathogen in soils. So the real test comes from whether you can relate the results of that DNA test to the amount of basal rot. And there's a lot of noise in these graphs when we tried to do that. So from each of those 21 sites, we actually, so the previous graph was, if you like, an average for each site, but from each of those 21 sites, we had 6 areas, and each area was monitored for disease at the end, at harvest, after a period of time in store. So here we see graphs, and we just divided them into 2 for a bit more clarity of the amount of pathogen DNA that we detect on the y-axis and the percentage bulbs that went on to develop Fusarium. As you see, there's a bit of noise there, but there is some sort of correlation between the two, although you'll notice also we do have some instances where we don't seem to detect the pathogen in the soil, but you still get a few bulbs with Fusarium, and the opposite is true as well. So we are, again, we're doing this for 40 fields this year, so we'll have some more data to look at. And what we'll need to do in the future, just try and pick out those fields that we feel where we're not detecting but disease does develop, to understand why we aren't detecting the pathogen in those fields. So the other part of that FUSE project I mentioned, I'm just going to talk about this for a couple of slides, was this idea of volatile gas detection to detect infected bulbs in store. So this was work that I did with James Covington. So he's an expert on electronic nose and various GC-MS approaches. And so we began very simply by putting healthy bulbs and infected bulbs in boxes and analysing the gases. So this is kind of James's territory. We got a couple of publications out of it, but you can see from the graph there that we could distinguish Fusarium infected bulbs from healthy bulbs, and James could also identify particular gas markers that were related to Fusarium basal rot infection. So within these electronic nose systems, there are different sensors, and you can then go on to test those sensors as to whether they can distinguish healthy from infected bulbs, knowing something about those gas biomarkers. So within the FUSE project, that's what we did. And here's a very crude setup that we set up here at Wellesbourne. But basically, those black containers contain healthy onions mixed with different amounts of infected onions. And then James kind of rigged up his gas sensors, testing different sensors for their reaction to these different amounts of infected onions. So these graphs just show the sensor responses, but the take-home message from this, that yes, we can identify sensors that do respond to the presence of infected onions. So he has taken that information and he's put the key sensors into this blue box here, and this blue box you can now deploy and is being deployed into stores. And here's an output from one of the sensors to show the difference between the healthy and diseased response. So these currently have been tested in experimental stores at ADAS, but the next step this year is this box will be going to commercial stores, with the idea particularly whether we can pick up the development of Fusarium during the curing and drying process. So hopefully more results will come on that later on in the project. Okay, for the second half then, or the last half of my talk, I'm going to switch then to Fusarium wilt of lettuce. And some of the work we've been doing on resistance, and particularly the characterization of potential new races in Europe and also in the United States. So a bit of background then. I mean, how do we identify these different races? So if we start with an infected lettuce sample, then obviously there's characteristic symptoms, which are characteristic of Fusarium lettuce. As you can see here, this vascular browning is very typical alongside the wilting of the lettuce plant. And what we can do then to begin to understand what race might be present in this lettuce is firstly we can do some housekeeping gene sequence, and then that confirms it's Fusarium oxysporum. But then there are also published PCRs for both race 1 and race 4. So we can, if you like, identify those key races if they're present. And then if you remember back to my diagram about 6 genes, we can also do some 6 gene profiling, as we call it, because we know that race 1 contains 6, 9 and 14, and race 4 contains 6, 8 in addition to 6, 9 and 14. So those are some of the things we can do, at least to distinguish 1 and 4, which are the major races that we're dealing with in Europe at least. Then a key part of identifying races is also to test against a differential set of lettuce cultivars, and there's a diagram on the right that represents that. So for instance, if we look at Costa Rica number 4, halfway down this table, you can see it's highly resistant to race 1 and susceptible to race 4. So that would be a key differential for distinguishing race 1 and race 4. Similarly, Lomaria here is susceptible to race 1 and highly resistant to race 4. So phenotyping also an important part of confirming race identification. So in the UK, as I said, we have race 4 primarily, but we've picked up a bit of race 1. And just a reminder that spread to the field is possible, certainly, or if may even be likely in the UK in the future. So spread to the field has been picked up in some parts of Northern Europe already. So my big shouty capitals there, be vigilant. Okay, so we had a couple of PhD projects where we were looking at profiling then these different foliar isolates that were coming out of Europe, because we wanted to know, you know, what the prevalence was of race 4 or race 1. We also have great collaborators in the USA as well, so we were doing some profiling of races from, of isolates from America as well. So as I said before, a number of columns here which can help you distinguish the different races. The first one is the translation elongation factor. We can put them into different groups depending on the sequence. And here you'll notice, in fact, the translation elongation factor sequence will distinguish race 2 and race 3. But race 1 and race 4 are in the same group. They're in group 1. Then we have the specific PCRs for both race 1 and race 4. And then, as I said earlier, we can look for the presence of the 3 key 6 genes, 6'8", 6'9", and 6'14". So you'll see that race 2 and race 3 also contain 6'8", as does race 4, whereas race 1 doesn't. But while doing profiling of these various isolates that we were receiving, a couple of things came up which seemed anomalous, alongside some of the phenotyping that was being done by industry. So I'm trying to highlight some of these in the table. So the first couple of variants that we came across were these, what we're calling race 1 variants. So they kind of look like race 1 in terms of their test sequence. They're amplified with the race 1 PCRs. But there was something funny going on around 6'14". At first, we couldn't detect 6'14" in these particular variants because the primers didn't work. But so we thought it wasn't there. but then the internal primer suggested 614 was there. So there's something going on around that locus that was different. And these isolates were known to break the differential resistance cultivar Costa Rica. So these Costa Rica breaking variants. We also saw some variants that looked identical to race 1 in terms of the profiling that also broke Costa Rica. So in this case, the profiling is not really telling us much about what the nature of these variants are or why they are breaking resistance. We also saw some race 4 variants. So one of these from Belgium, you can see the second one down in this little highlighted group of 2 didn't contain 6.8, but yet it wasn't race 1. So something funny going on there. The other one looked pretty identical to race 4, but did break a race 4 source of resistance. And finally, probably the most interesting to us was an isolate from France, which had a completely different test sequence to anything else that we'd seen before. It didn't amplify with either the race 1 or the race 4 primers, and it had a different 6.8 sequence as well, and it seemed to break multiple sources of resistance. So that to us looks like a new race. But we're currently, again, in collaboration with Helen Jordan, carrying out some genome sequencing to sort of investigate these variants a bit more closely. But just to illustrate that in the form of some phylogenies here, here's the translation elongation factor phylogeny. So this is based on sequence. You can see all the race 1, the FOLA 1 and FOLA 4 isolates clustered together. And we can distinguish, in this case, race 2 and race 3 quite nicely in different clades. But this new race, of which we have several exemplars from France, occupy a completely different part of the tree. Similarly, for the 6.8 phylogeny, again based on the 6.8 sequence, we see that race 2, race 3, and race 4, the 6.8 sequence clusters together. We also found some race 4 isolates have a slightly different 6.8 sequence, but again, these French isolates are on their own. So we think this is good evidence that they are something quite different. So Emily Turner, one of my PhD students, has done some phenotyping on some of these variants with a focus on the race 1 variants. And this is just to illustrate the breaking of resistance that I mentioned earlier. So here we have a race 1 variant here, and you can see this just shows the amount of disease at harvest, so the vascular browning score, and it shows Costa Rica here becomes highly infected with this race 1 variant compared to the race 1 isolate on the left-hand side here. And then for the new race, or what we think is a new race, then this is breaking multiple sources of resistance. So all these different differentials seem to get infected, some of which are at a slightly lower level than for some of the other races. Okay, so the challenge then is to develop new sources of resistance to these new races. So that's something that the industry is going to be addressing soon. But our focus when we started this work was in developing new sources of resistance to race 4, because that was the newly emerged race in the UK and Europe. And we had some good resource to do that. For instance, this is a picture of an infected polytunnel, and you can see some nice exemplars there of some resistant accessions and some diseased accessions as well. So part of this work was done as part of the Vegetable Genetic Improvement Project funded by DEFRA, and we screened the whole diversity set that we have from the gene bank here at Warwick, and we identified resistance to both race 1 and race 4. And then in a PhD project with Andrew Legg, we had a collaboration with Enza Zarden, where we were developing resistance also through production of mapping populations. So that would be for future genetic analysis. So here's some example then of results from our race 1 and race 4 resistant screening. There's around 90 odd accessions here, lettuce accessions along the bottom axis there. And you can see the vascular Browning score, so the severity of the disease on the y-axis. So you can see a good range of reactions there and differential reactions for some of these lettuce lines between race 4 and race 1. And I've divided this into different lettuce types, and you can see the butterhead types are on the left-hand side here, largely are very susceptible to both race 1 and race 4, so there's not good sources of resistance there. But for other types, such as the Batavian on the far left side, we've got a couple of accessions there that are resistant to both races. Interestingly, some of the leaf cutting varieties here seem quite susceptible to race 1, but are resistant to race 4. So there is new sources of resistance out there in this lettuce diversity set. So the same could be done with some of these new variants that are currently emerging. So with Enzazardan, we went to test some mapping population parents in comparison with commercial lines. So this graph just shows you a number of different resistant lines, which we picked out from the diversity set, which we confirmed the resistance in, so very low vascular Browning scores, compared to some susceptible parents there of mapping populations. And some of the early commercial material here, we did get some infection with that, so we would say that that was partially resistant, but I think that has also improved more recently. And finally, just some photos, just to show you the nature of this resistance on the left-hand side. Here we've got exemplars where we have uninoculated plants for race 1 and race 4, and their corresponding resistance counterpart. So basically in an inoculated system, we see a pretty much identical phenotype then in terms of resistance compared to the uninoculated. On the right-hand side there, you can clearly see a race 1 inoculated plant that isn't resistant compared to the uninoculated, and a race 4 inoculated and uninoculated. So we do see some strong levels of resistance in this germplasm. Okay, so one question that also emerged was, does race 4, can it still colonize resistant lettuce plants? So Andrew Legg, my PhD student, went out to test this in a fairly simple experiment. So here we have a number of different lettuce lines along the bottom. Some of them are resistant and some of them are susceptible, as you can see with the vascular Browning scores on the left-hand side. And then what he did was just see whether he could isolate the pathogen from the main taproot of the lettuce plant in either these resistant or susceptible varieties. And the results were a little surprising, actually, in that even in the resistant types, which you can see on the left-hand side, we could still isolate the pathogen, certainly from the bottom of that taproot, but also in the middle to a less extent in the top, whereas colonization was more extensive in the susceptible varieties. So we think it is the case that sometimes these lettuce plants can look completely asymptomatic, but the pathogen may still be present. So that does raise the question of, if you plant a resistant lettuce, does it actually result in a reduction in inoculum? And finally, which sort of follows on from that, we were sort of asked the question by growers in particular as to whether non-host crops could be colonized by Fusarium. And here's a couple of experiments we did with FOC on the left-hand side and race 4 of the lettuce Fusarium on the right-hand side. And these are quantitative PCR results of colonization of these pathogens on roots of different plants. So you can see for FOC on the left-hand side here, we can see a number of different crops are colonised, albeit at quite a low level, compared to onions, where you'd expect a high rate of colonisation. And on the right-hand side, for the lettuce pathogen, again, we can see that some of these other leafy crops are also colonised by the pathogen. And of course, we get a much higher signal here for susceptible lettuce types. So again, that does raise the question about how efficient rotation plants might be, again, in reducing inoculum if some of those root systems can be colonised by these Fusarium pathogens. Okay, and as a final postscript, just say I've obviously focused the resistance work on lettuce, but just to flag up that we did do some work on looking for resistance to Fusarium oxysporum CP in the past in collaboration with Hazira, but I'm not going to say more than that today. So I've taken up much of your time, but there's quite a few people to thank. So within my group, Sasha Jenkins here, been doing much of the FOC diagnostics, and Emily and Andrew I've mentioned, doing the profiling of those foliar isolates. Jamie from Murray Grant's group, he was helping to run the bioinformatic pipelines, particularly for effector discovery, the pathogenicity gene discovery, and particularly Helen and Jordan at NIAB, as I say, long-term collaborators. They're experts on nanopore sequencing, the long-read sequencing that's been necessary, and to run those pipelines. I've also got great collaborators in the US, so Cassandra Sweat and a consortium that's been put together for a grant proposal based around UC Davis, and Kelly Richardson on the lettuce Fusarium, part of the lettuce Fusarium group that I collaborate with at USDA. Lots of people in the Fused consortium, which I can't mention them all individually, but thank you all, and also these various funders for providing the funding. So thank you for your time, and very happy to answer any questions.
Brilliant, John. Thank you very much indeed. So I think there's a comment and also a question in the chat. And then if anybody else would like to ask a question, then please put your virtual hand up. So do you want to have a go at the question in the chat, John?
Yes, so that's an excellent question, and a bit of information I didn't impart. So from Kelly, so it's a diagnostic assay based on a particular 6 gene. What about the other formae speciales that also have this 6 gene? Well, conveniently, it turns out that the sequence of these 6 genes between the different formae speciales can be quite different. I neglected to tell you that vital piece of information, which means those differences are still enough in some cases to develop a specific PCR assay. So thank you for that question. That's hopefully cleared that up for a number of you.
Yep, I don't know whether there was a comment from Latvia about the above
70% of onion harvest lost to Fusarium. Wow. Okay.
So yeah, quite high levels. Right. Does anybody else want to ask a question? If
Have
so,
to be brave. Everybody can hear you.
Yeah, yeah. Well, you can type it in the chat if you want.
Yes, that's true.
No. Oh yeah, here we are. So there's hang on, there's the chat again. Any potential chemical control in brassica?
Chemical control in brassica. I think yes. We've not looked at brassica Fusarium in particular, although we're beginning to now. I think as a general rule, fungicide control is very difficult for Fusarium diseases because they're soil-borne. Certainly, when we've become involved in testing in the UK, then the azole fungicides tend to be more efficient against Fusarium. But of course, you've just got an application problem, basically, and I think you'd have to sort of steer between the label advice in terms of applications. But early application would have to be something that would have to be done to have any kind of chance of control. But really, it's not very efficient.
Okay, and Jordan's got a question. Would you like to ask the question, Jordan?
He should know all the answers,
Yes.
Jordan.
Well, it's about the FOC. Well, it's a good talk. Thank you. So I was just wondering. So in the diagnostic test, where you had infection in the onions where there wasn't any Fusarium DNA detected, do you think they represent unidentified FOC isolates or something? Or is it a problem with the test?
I think what's happening here is that so these are asymptomatic bulbs. So I believe that they are infected with Fusarium, but not to a sufficient level where you can see symptoms. Yeah. So, and that is where I think what we really don't understand is these kind of late infections. So they're not causing disease directly in the field, but maybe there's something around harvest time. Somehow the pathogen's getting into that basal plate around harvest time. And then, of course, during the curing and drying process, you're blowing hot air at 28 degrees through a massive stack of onions, which at least to start with are quite moist. And I think that actually gives the conditions which are excellent for any latent infections to then develop. Yeah. And I think that's why the kind of volatile detection is becoming more focused on that phase, rather than when those onions are subsequently cooled and held at 5 degrees, because we know that the Fusarium really doesn't move much at 5 degrees. Yeah. So I think it's these latent infections that are causing the problem, and I'm not sure we can't understand how or when they occur, but it's a problem for the industry.
Thank you.
Excellent. Right. Any more questions before we close? Oh, another one in the chat.
Are there other pathogenicity related genes apart from 6 genes commonly used to characterize races? Commonly used, maybe not, but as Jordan might probably tell you better than I, there are plenty of other pathogenicity genes which we can identify by bioinformatics in these Fusarium genomes. I think as researchers, and just because of the nature of publications, everyone got a bit obsessed with these 6 genes, but we have to remember that these 6 genes are only kind of a subset of potentially a much larger set of pathogenicity genes that are present. So there's other pathogenicity genes, effector genes, to be discovered and characterized. So there's no reason why some of these unknown genes, if we find they are unique, couldn't be used to characterize forma specialis or races.
Great, thank you. Right, looks like there aren't any more questions. So thank you everybody for joining us this afternoon, and a big thank you to John for loads and loads of information. Fantastic.
Yes, I'm happy for anybody to email me after with any further questions. Yeah, that would be great.
Brilliant. Thank you very much, and I'll send round the link to the recording once it's available, and also something about the basis and Neroso points. So thanks again, and have a good evening.